Welcome to everybody. I never quite thought about the fact that I've actually worked for two companies that don't exist anymore, but of course Parke-Davis never ceased to exist, it just got bought by Pfizer. But so I don't know if it was by design or just fortunate that my talk is sort of going to touch a little bit on some of the things that Peter talked about before, and certainly about the AssayMap technology that Scott talked about, and it's sort of my personal experience in these different things. So first of all, I'm just going to start with a little about my background real quick. Like Mark said, 15 years of experience. I started out at Parke-Davis supporting the high throughput screening lab, and that switched over to Pfizer, and it's all small molecule, large compound libraries, and all the problems that go along with that. So dealing with data handling, dealing with automation issues, and library prep, and all the screening that goes with that. The data side of it actually was the biggest challenge, and I spent quite a lot of time doing software development for tools for that, for those sorts of activities, and getting the data back out to the project teams that needed it. I left Ann Arbor and moved to Seattle and started to work for Rosetta, which was part of Merck. And there it was more of a process engineering sort of environment where we were taking protocols that were already in place and trying to improve them and make things more efficient and get a little bit more out of what we were trying to do. So it was sort of a little different approach to the automation. In 2009, Novo Nordisk, which is primarily a European company, it's a Danish company, decided that they wanted to open up a research site in the United States, and they chose Seattle. And it's not only a new research site and a new project for them, but it's actually a brand new therapeutic area. Novo Nordisk is known for diabetes research and insulins and delivery systems, but in Seattle, we're primarily focused on autoimmune diseases, and primarily rheumatoid arthritis, Crohn's disease, IBS, multiple sclerosis to an extent. But it was an opportunity for me to take the experiences that I've had in automation and apply them to a completely blank slate. Basically, it was a brand new site. There was nothing in place. All they knew is they wanted automation. It was completely open-ended, and that's sort of an automation person's dream, to walk into a situation where you can do whatever you want to do, and as long as you make it work, everybody's happy. So that was sort of what I stepped into in 2009, and the first thing they said to me is, we want an integrated system that can do tissue culture work. Well, that's pretty open-ended. That didn't really give me any direction on what they really wanted to do, and I said, well, I need a protocol to work with. Give me some idea, give me some place to start. Well, I sat down with our Phage display group, and who coincidentally was going to need the largest amount of automation. And the head of the Phage display group and I decided that a good place to start would be 24-well plate transfections. So the idea was, and I'll get into this a little later, but once you've gone through the whole selection process on a Phage display, you're going to have X number of clones that we wanted to put into mammalian cells and convert to IgGs. And in order to do this, we needed to bulk up the volumes to a range where we would get the kinds of yields that we would actually be useful to us for downstream processes. So we decided that we were going to try to tackle 24-well plate transfections. To do that, we knew we needed an incubator, something that would handle the cells and keep them happy. We would need at least one liquid handler, possibly two, because we have 96-well plate work, and individual channels because we would have to reformat to the 24-well plates. We also needed a sterile environment and the plate and tip capacity to be able to handle the throughput that we desired. And so we also said, well, if we're going to do this, we're going to spend the money, we're going to buy a system, we need to be able to do a lot more than this. And I'm really big on flexibility in the automation, and supporting a discovery system or a discovery site our projects are changing from year to year, even within the same year. We change targets all the time. We're looking for new and different and innovative things all the time, and in order to support that from an automation standpoint, you need to be able to handle lots of different things. And so we also sat down and made a list of the types of equipment that we might want and we might need, and of course it was much longer list than was actually possible to ever put onto any single system. But we went out and we talked to companies, and we looked around, and we decided that Agilent was going to be the way to go. And once we pared down our list to a reasonable level, we came to our first BioCel. I don't know how easy it is to read all this, but this basically has an incubator underneath. This is a BioCel 1200, so it's the medium-sized BioCel. But we have a Bravo liquid handler with a 96 well head. We have a Hamilton Starlet, which has a 96 well head and also 8 individual channels. Our incubator is underneath the platform, and it feeds plates up to the top. And we have the plate hub for plates, and the stackers for which we use to introduce the tips. Now, since there is still room on here, we put 2 biotech plate washers, the EL406s, which have both plate washing capacity, and they both have syringe pump dispensers and peristaltic pump dispensers. So we have a lot of dispensing capability. And then the centrifuge, which we ended up using for lots of different things. There's also a plate hotel and a barcode reader and a lid hotel, which just gives us added ability. But this was our basic system to start with. There's a couple pictures of it. You can see the Hamilton and the plate hub. Centrifuge is in there, and the stackers. And this is sort of the front side. It's a little bright with the light. But that's the Hamilton. You can see the incubator underneath, and the plate washers in the Bravo. So this is what we started out with. I want to talk about phage display real briefly, just so if people are unfamiliar, you kind of understand the process. And I'm a chemist, and I do automation, so if I don't get all this stuff quite right, forgive me. I don't know if we have biologists in here or not, but basically you start with a pool of like a tube of that contains your entire phage library, the entire diversity of it. It's about, I think this library in particular is 1 times 10 to the 7th, I believe, diversity. And you basically introduce that to an immobilized antigen. Whoops. Now, the antigen can be immobilized on any surface. So you could use a microplate. You could use an immunotube. It could be a cell-based antigen if you have a cell line that overexpresses a particular target or protein. You could use beads. We use beads with the proteins immobilized on them. But anything that can present an antigen, and then the phage can actually bind to. And you introduce them. You mix them together, and what you get is you have your specific binders, which is shown here in blue, which will bind to it. You'll get some measure of non-specific binding, either binds to the protein in a way that you don't expect, or it binds to the surface somehow. And then you have all the other phage that won't actually bind to your particular target. And then you wash them away, and what you're left with is these that are binders. You infect E. coli with them, and you grow the cultures up, and you harvest those, and you put them back through the process again. And the idea is every time you go through the selection process, you get a more and more enriched group of antibodies that are specific to your particular antigen. And so you get, theoretically, you get a better sort of hit rate out of this. What you end up losing as you go through these selection processes is your diversity. So it's sort of it's a balance between one and the other. But once you've gone through enough selection processes, you actually get this out, and you get your antibodies out in a soluble form, and you can run some sort of an assay on them. Now, you might run the assay after selection 2 and 3 and 4 or whatever it may be, but but ultimately you're running some sort of ELISA assay or flow assay or something, some sort of quick answer that will tell you if you have a binder or not to your particular antigen. Now the idea is once you have these hits selected and you've got a set of plates, generally each one of these selections will result in maybe 4 to 5 plates, and you may do 3 or 4 or 5 different types of selections for a particular project or target. You may end up with 16 to 20 plates. And the next step at that point is to go on into the IgG conversions, and this is where the automation comes in. So our first protocol was this 24-well transfection, and it's basically a normal transfection reaction. Once you grow up your final selection cultures, you harvest them and do a plasmid prep. You take the DNA from that plasmid prep and you go into this reaction or this transfection reaction. Combined with media and the Fectin reagent, we do that on the Bravo. The plates are incubated in the plate hotel for about 20 to 25 minutes, and then they get combined with the cells on the Hamilton. The cells sit in the incubator until they're ready to come out. They go out to the Hamilton. Everything's combined. They get put away, and then the process repeats. We've run everything from 1 plate through this process all the way up to 20. The protocol is currently designed to handle up to 20 at a time. But it really depends on how many plates we have to run and what else we have going on in the system that particular day. But once the protocol is finished, all the cells go into a shaking incubator for 5 days. The second part, after the 5 days, is they come out and they get harvested. So the cells go back to the Hamilton. We harvest the soups into a deep well block, spin that down, and then it goes to the Bravo, and we take a certain amount off the top. All the plates get put away, and that process repeats. So basically, when all said and done, we have 96 well plates with our IgGs from the original DNA. So what we did is we did some initial experiments, and the first thing we wanted to do was compare our automated transfection versus the manual. And there's a couple of different things in this slide. These 4 columns on the left are 4 24-well plates, and this is basically, we just took the same clone, it was sort of an average yielding clone from our process, and we pulled the samples and checked the yields. We got between 50 and 60 micrograms total per well, actually, for the automated portion. The manual portion these 3 plates were all done manually, and the difference here is this one had the DNA and the cells combined, and then immediately went into the shaking incubator. This one set for 3 hours before it went into the shaker, and this one was 6. And we were concerned about the time spent between the first plate in a long automated run and the last one, which conceivably would be 20 plates later. That first plate would sit there for approximately 6 hours. And so we wanted to make sure we weren't going to take any hits in yield by letting those plates sit on the system for a time. And you can see that there really isn't much of a drop-off. In between 0 and 3 hours, there's hardly any notice at all. And the difference in yield between the automated and the manual process is negligible for our purposes. And this also, because it was the first run, we learned a lot between then and now, and some of this loss is actually due to evaporation. We've corrected those issues. Another thing that we discovered was, and this was quite by accident, but we took just an average plate that had a bunch of different sequences out of a selection, and we ran them through the process, and we wanted to get an idea of what different types of yields, the sort of a range of yields we could expect. And what we didn't know is we had a number of wells on there that had the exact same sequences in them. And it turned out that the wells that have the same sequences actually came out with pretty reproducible expression levels. So these blue ones are all the same sequence. They all came out to be about 12. The 2 yellow ones were about 24, and the red ones were about 62, 63. And so we felt really good about the fact that this was pretty reproducible based on expression levels according to sequence. You'll also notice down here, I don't know if you can see it, but there's a lot of zeros in here. And it turns out that a lot of the sequences have stop codons in them and they just don't express well. And since then we've put in a sequencing triage step to take those out, so we're not actually transfecting a lot of wells that aren't going to produce. But this was sort of an accidental discovery on our part and was very encouraging. Another thing we did is we wanted to look at different formats because the 24 well plates produce really well, but it requires sending 5 separate plates out to the Hamilton. And on our BioCel, the Hamilton itself is sort of the bottleneck, not the Hamilton, but the transition of plates from the BioCel out to the Hamilton and back again is a bit of a bottleneck because we can only move 1 plate at a time. And so we figured if we could switch to 48, we could cut down to 3 plates instead of 5. And 96 would be even better. We wouldn't even have to go to the Hamilton. But you can see from the yields that the 24 well, this is actually a very high expressing clone. It's 150 micrograms. The 48 well is down around 90, and the 96 well is down around 10. We put the 30 in here just to kind of show that this is sort of a standard 30 mil transfection. A manual reaction will get you about 200. We're getting about 150 from a 24 well. But you can see the 48 well is pretty good, and it actually saves us some time. So the majority of our transfections now are done in 48. So the numbers, we have the capacity to process 5,000 clones a week, working within a normal 8-hour day. For 10 DNA plates, it's about 3.5 hours for the transfection method and 2.5 for the harvest. It turns out there's only 1 day a week where we can do both on the same day as a Wednesday because of the 5-day incubation. And nobody wants to come in and work on weekends, certainly not me. But we tend to do transfections Wednesday, Thursday, Friday, and we harvest Monday, Tuesday, Wednesday. The reality of it is we haven't had a need to do this many clones in a week because we still are in the process of putting the upstream and downstream processes in place to handle it. We just don't have the upstream processing capacity to have a need to do more than that. But we can if we have to. And so to date, we've been using this method this protocols for just over a year, and we've produced over 30,000 clones. The nice thing about these 30,000 clones is they're all very target-based. So it's not just 30,000 random clones. They're actually specific to targets that we've been working on for particular projects. So we've been able to get a lot of useful information out of these, and it's something we never would have been able to accomplish without the automation that we put into place. So real briefly, you'll notice on the first BioCel that we didn't have any kind of reading capabilities. We can set up assays and we can get large numbers of plates ready to be read, but we had no way to actually read them in a high throughput manner. So we put this second BioCel together. It's actually sitting in the same room. It has a high throughput flow cytometer on it. This is the IntelliSite system. And we've done up to 30 plates in a single run on this system. We have an in-cell imager. We also have an Envision plate reader. And between these 3 instruments, we have the ability to do almost all of our assays in throughput. Probably 90% of what we've wanted to do so far we can do on this system. And the things that aren't on this system, we're working on automating those as well. There's a couple pictures. You can see there's the IntelliSite system there, and the Envision and the in-cells in the back. And this is just from the other side, so you can see the Envision and the DDR. So this leads to the next step. Our next need was high throughput protein purification. So what happens is after the transfection process, we get the IgGs out, and in most cases, if we're just going to ELISA, sometimes we're repeating an ELISA or flow assay that we ran upstream on the phage. Now we want to repeat that experiment on the IgGs and see if our results are similar, if if we have any stronger binders now. A lot of times we get better flow results from the IgGs than we did from the phage, and oftentimes it's just due to affinity issues. But for those types of assays, we don't need to purify. Where it comes into play is if we want to run functional screens. And for the functional screens, we really do need to have the purified IgGs. We have a protein purification group on site, but they deal with larger volume proteins on a smaller scale, a smaller number scale. So they can't really handle the throughput that the phage display group needs. And they don't really want to deal with the volumes and the concentrations that we're dealing with. So we decided we needed some sort of system to be able to do this. And we started looking at it, I would say, late 2009 or early 2010. And we decided what do we need? We wanted to be able to purify up to 5 mils of IgG or Fab supernatants that coming out of these transfections, and with a minimum output of 10 micrograms. It's a pretty low target for our yields, and we thought that would be a good place to start, and it would give us more than enough material for what we needed. We wanted to be plate-based, 96 well format preferably, and to be truly high throughput. Endotoxin-free. Most of our functional screens can't tolerate endotoxin, and reasonably inexpensive. We found in this process that protein purification is not cheap, and so we were hoping to find something that we could feel comfortable with if we were going to go to large numbers of plates. So we tried a number of different things. We tried tips, plates, and magnetic bead-based. The costs, I say reasonably inexpensive. That to me is reasonably inexpensive. So about $150 this is per 96 well plate, so per 96 samples. The tips are obviously the most cost effective. The plates, $750, magnetic beads, $1,200 are obviously very expensive. The percent recoveries in all three of these methods were not great. 20 to 30% for the beads and the tips, and only about 10 to 15 from the plates. Now, the caveat to this is we didn't put a lot of time and effort into optimizing these. We took a look at them, we tried them out, just sort of rough methods, and to see just kind of where we were at as a baseline, and this is what we got. The tips are automatable. The plates were not really. It's not really a protocol that's easily automated. You would think 96 well plates would be, but the way that they were designed to be used, it wasn't something that we felt we could automate well. And the beads, it kind of depends on your approach. We were really looking at a Kingfisher system, which is sort of a semi-automatable. There's other ways to do it, and there's probably ways we could find, but volume-wise with the magnetic beads, we thought we were going to be too high to be able to automate it very easily. And the other thing we found on the tips and the beads is we really needed, this actually should be 20 micrograms per mil. Anything we had that was less than 20 micrograms per mil was sort of washing out of our purification. We weren't getting any kind of detectable yield out of it if we started with concentrations that were that low. And it was kind of troubling. We tried a lot of different things to get around this, and we really kept running into that barrier, whatever it might be. So we were sort of at a loss. We didn't have anything that we really liked, and when I went to lab automation this year, one of my goals was to find something that might be better than this. Fortunately, Agilent announced AssayMap at Lab Auto this year. And this is an Agilent slide, by the way. Actually, I think it came from Scott's presentation at Lab Auto. But and what we ended up doing is talking with Agilent, and we decided we were going to go ahead and give this technology a try. So we got the Bravo head, and we got the cartridges, and we decided to take a stab at it. The first one we did was really minimal optimization. It was just like the other runs we did with the other methods. And we took a single clone over in all 96 wells of 2 plates. So we had 300 microliters total, 150 microliters in each plate. And the method is pretty simple, and it's sort of, Scott sort of described this, but we equilibrated the cartridges. There were 2 separate loading events. So it would load from the first plate, drop the cartridges, empty out the soup, and then do a second load, a wash step, elution, a neutralization step, and then we have to have a strip step in our purification, because our IgGs actually have a VH323 framework, which contains an additional protein A binding site, which means our IgGs are really, really, really sticky. They're really hard to get off, and we can elute most of it off, but it's tricky to get the rest off. So we actually have a strip step in here, and then we re-equilibrate, and the cartridges are ready to go again. So this is what we did, and we did our quantification on an octet red. Our average input per well is 62 micrograms. Our average output was 52 micrograms, plus or minus 3. This is an N of 62. We got a percent recovery of 84%. Now, this is a non-optimized protocol, and we were ecstatic with this. I mean, you saw the other recoveries, we were getting 20 to 30%, and we got 84% right off the bat, and we were pretty sure at this point that we had our path forward for purification. So another experiment we did was head-to-head with magnetic beads. This one we have 7 different clones, and we did a bead-based purification with a 5 mil input, and then we also used the AssayMap with a 1.8 mil input. So the input from a microgram this is in micrograms. It's almost 3 times the amount of input that we put into the AssayMap cartridges, and in all but one case, the output was actually higher from the AssayMap cartridges. And you can see that it reflects in the percent recovery, 18% average for the beads and 67% from the AssayMap cartridges. And also, note this is post-buffer exchange, so there was definitely some lost in both cases because of the buffer exchange process. You just always lose some in that. And I think prior to the buffer exchange, we were around, I'd say, between 75 and 80, and probably around 25% for the beads. But the fact that we can cut our input down to less than half and actually get the same amount out is very telling. We also looked at our low input issue. We started with 20 micrograms, went down to 5, and even at 5 microgram input, we got over 3 out on average. So really, as long as we can get something back that we can throw into an assay, we're pretty happy with that, and this is really really nice. And this is also, again, without really optimizing the protocol. The last thing we wanted to look at was a cartridge use limit, and really what we wanted to do was see 2 things here. If our yield sort of stayed the same, if we used the cartridges over and over, we also wanted to see if there was any kind of carryover. And so what we did is we took a clone across an entire plate. I think it was around, I'd say 18 to 19 micrograms of input. And we run 4 plates back to back, and then the 5th plate was just media in it, and no IgG. And we basically just wanted to see that we had a similar recovery across all those runs, and that we got nothing in every 5th plate, and that's basically what we got. I also want to point out that so this made us comfortable that we might be able to run them up to 15 times. But of course, this is particular to this protocol and these actual steps. If you change things around, this might not quite work the same. These also were run pretty much over a 2-day period, so it's not like we ran a couple and then it's set for a few days and then we ran it again, and these numbers may change slightly because of that. But what this told us is we could be pretty confident that we could run up to 10 and maybe even a little higher, and still get reliable results out of them. So price-wise, it's definitely reasonable. I mean, we're down in that range where we're comfortable around $170 per 96 wells, and that's, I think I calculated that on about 12 uses. So we're somewhere in that range. Automation, it's a fully automated platform. I mean, it's the head is on the Bravo. The Bravo's on the BioCel. It can't be any more automated than that. So we're, the only issue that we need to work out is refilling our buffers, but I've got some solutions for that. The input volumes, we've gone up to 200 microliters per load. We can go up to 250. For some of the samples that we've done, we've actually split them across an entire row, so that we would, and then pooled them back together afterwards, which is fine if you don't mind having a little bit more dilute material, because you're basically multiplying your elution volumes that way. But we are working on optimizing multiple loading protocols where we would just load the same samples multiple times, and that seems to be working pretty well. Endotoxin, I didn't really talk about it, but we did do testing on endotoxin from the cartridges, and there is none. We have had some endotoxin issues in our lab, and a little from the process, but it's not coming from the cartridges, and we think we've narrowed it down to some of our own things. So as far as the cartridges go, we're not worried about it. And the recovery is excellent. I mean, we get around 80% consistently. The next thing we're going to do is play with flow rates a little bit to try to get that up a little higher. But honestly, for our usage, we're way over what we need to be at, and we're pretty happy with that at 80%. So that's it. And thank you all for listening. And if anybody has any questions, feel free.